G'day guys, this is on short chain fatty acids from protein sources. So let's get into it. I'll just share my screen. So this is just I'm using this other study here, which it's a review, so it's irrelevant to what we're doing. It's just to basically cover a point in this. So I'll just read part of it. Short chain fatty acids are small organic monocarboxyl acids with a chain length of up to 6 carbons. Obviously, they've got some, like a hydroxyl group and a sort of another ring, which is a carbon hydrogen sort of ring. But anyway, we won't get into chemistry right now. And the main products of anaerobic fermentation in ingestion of polysaccharides and polypeptides as well. So these are long chains of sugars coming from dietary fiber and resistant starch, in this case produced by the microbiota in the large intestine. So, when Bart says all carbs, even dietary fiber and resistant starch, they all turn into sugar. Exactly, because that's what they get fermented. So these polysaccharides, long chains of glucose, can actually be utilized by these little buggers. Comprised mostly of, comprised mostly of acetate. You do get more acetate when you're deriving your sources. So the bacteria will produce more acetate from dietary fiber and resistant starch than they will from proteins. That is true. And acetate can actually go back to the liver and get converted to glucose. So more glucose under certain circumstances. Propionate and butyrate, which is the big ones that everybody talks about. But anyway, there's also butyrate and a number of others that are very important as well. And with the body being a Swiss Army knife that it is, with all the different enzymes, it can basically do all that little trickery in the body. It's a marvelous system. In an approximate molar rate of 60, 20, 20, respectively, approximately, and this is the information that I wanted, 500 to 600 millimoles of short chain fatty acid are produced in the gut per day, depending on fiber content, blah, 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 transit time, yes, all that sort of wonderful stuff. So basically, that's the sort of number approximately that gets produced in general population. Okay, let's move on. This is a study that I wanted to show you, degradation of amino acids to short chain fatty acids in humans, an in vitro study. So this was done by the Department of Medical Gastroenterology this was at Hyvidorvi Hospital. I've probably killed the pronunciation of that. I think that's a town close to the Swiss borders from memory. University of Copenhagen in Denmark. Okay, and so that was basically published in the Scandinavian Journal of Gastroenterology in 1988. We go right to the bottom. That's when it was received. So it was received on that date and accepted on the 5th of October, 87. And then by the time it went through peer review, it got published in that periodical the following year. Anyway, let's move on. So short chain fatty acids originate mainly from the colon through bacterial fermentation of polysaccharides, which was the traditional view, the fiber, starch, resistant starch, things like, well, even back then, they probably didn't even really know as much about resistant starch. This was back in the 80s. The knowledge was more that, oh, it's all polysaccharides, from fiber, these lengths of glucose, basically, to test the hypothesis that short-chain fatty acids may originate from polypeptides. So polypeptides are a long chain of amino acids as well. The production of these acids from albumin and specific amino acids was examined in the fecal incubation system. Albumin was converted to all the short-chain fatty acids. So albumin is produced as a protein produced basically in your liver, and it constitutes about 4% from memory of your blood. And what it does is it prevents, one of the key things is that, I know a lot of people talk about, oh, it moves around, hormones and vitamins and enzymes in the blood, some other, anyway, from memory, and but the key thing that I always remember it for, because with my cardiovascular stuff, it prevents leakiness of fluids from the endothelium into the other organ systems. So if your albumin collapses, blood will seep into the body out of your blood vessels. Not a good thing. So a lot of people get worried when they get high albumin and get frantic. Don't worry. It's probably due to dehydration. That's one of the key things, or dysfunctional liver or kidneys that do the filtration, and so, but that's pretty rare. You'll know if you've got a problem with your liver or kidneys. You'll actually see a lot of yellowness, jaundice, yellowness in your eyes. So it's a bit of a giveaway. We carnivores don't see that, so we don't really have to worry. For most cases, if it's elevated, it's usually due to dehydration. So people doing a lot of exercise is one of the key things. It's contraindicated. What sort of tribal man and woman are you? Whereas amino acids generally were converted to specific short-chain fatty acids, most often through the combination of deamination and decarboxylation. So when deamination is like, it's getting rid of a hydroxyl group and adding in a proton. Yep. So it's just biochemistry. Who cares for this video? And decarboxylation, just generally, it's the nitrogen. So you're either removing it or putting it back or stuff like that, in a sense. So you put in, what they say is you put in place a carboxyl for some sort of amino combination of, but basically, it's really the nitrogen sort of removal or adding stuff like that. Anyway, of the amino acids, all through more complex processes also took place, but this is not a biochemistry class, and I struggle with biochemistry anyway. There's so many combinations. This study indicates that a part of the intestinal short-chain fatty acids may originate from polypeptides. This was some of the early studies being done in this specific field to determine this sort of stuff, so which apparently are a major source of those short-chain fatty acids is isobutyrate, threonate, and isoveronate. Only found in small amounts in a healthy colon. Moreover, gastrointestinal diseases resulting from increased proteinase material in the colon, exudation, mucosal decontamination, which is mucosal barrier sort of problems, bleeding, and so forth, may hypothetically influence short chain fatty acid production. Not hypothetically, they definitely do. So people that have got IBS and a lot of these sort of things do tend to have more problems in deranged gut functioning, which can affect the production of short chain fatty acids. How much, even to this day, it really varies between person and person and the gut microbiome sort of makeup and all that. So it's not an easy question to give. So I just don't answer it because we don't really have precision on that to answer it. It varies. It's very variable. So I'm not going to bother. So they got the stuff, the fecal stuff from a healthy male, 29-year-old who volunteered to give him a whole lot of shit. I couldn't help myself, you know me. And basically they then homogenized all these sort of mechanical changes so they could remove all these things. So then they could use that to basically put in the other and the actual amino acids, which they got from the Sigma company. And all these were done in, you know, these concentrations are in the milligrams. So these are very small concentrations. But remember what we saw before, 400 to 500 millimoles. You'll be surprised the sort of levels we're getting from basically like these small amounts, and from incubating, you're getting over 300, all the different types. So when we took, that's acetate, that large one. So we're still getting a certain level of acetate, quite a bit. That's one thing you get from short chain fatty acids, quite a bit, even from animal foods, even, you know, so even from basically those sources, it still happens. That's just a reality. But it's much higher in those who basically are on a plant-based type. Propionate and butyrate, our favorite, that white one there, quite a bit after 24 hours. And also the other ones, which is the isobutyrate, veronate, and isoveronate. So you get much less of that. So even with, you know, because a lot of what happens is a lot of the actual other studies that I've actually seen that are basically, the way they're done is not very good. I found that there's some of the modern studies that are deliberately done by vagoons to basically deliberately make out that you get more isobutyrate and you get very little butyrate. And as you can see, there's your butyrate. You know, C4, there's your butyrate. And there's your other ones, the so-called isobutyrate, veronate, and isoveronate, which are also fine because we've got enzymes to basically alter them back into butyrate. But it's just basically they always sort of hit us over the head by saying, oh, you produce a whole lot of, you know, isobutyrate and very little butyrate. Bullshit. We actually produce more. As you can see, that's up to 72 hours. You actually produce far more. And we know that protein actually moves through the whole system much slower, because you actually are adding to a pool of a protein pool continuously over many hours. So the more protein you eat, you'll digest it over many more hours, which you get satiety, but also you're giving these little fellas more time to basically deal with it, where most of the actual, you know, vagoons are shitting their pants every couple of hours, you know. So basically they are expelling contraindicated fiber and other things like that on a horrendous base every day. They don't even get it to stay in their gut for long enough. No wonder they're actually getting these lower quality quantities, where here you're only getting small milligram, you're not even getting to the grams, or basically, you know, in the large amounts that we actually consume in terms of aminos, like big steaks and stuff like that. So far more. So yeah. So we get plenty of short chain fatty acid. You don't have to worry one iota. in that regard. So let's move down and actually look at the sources, because this is really what interests me, is the sources. So where is it actually coming from? So when we're looking at acetate, you know, like these are, everyone is high in albumin, everyone. And we know if you're on a high protein diet, you'll produce more albumin anyway, which means that you will have better protected blood vessels, which is a good thing. And it also can be used for that purpose as well by, you know, so in the gut where the albumin is, it can actually, you know, some of these little buggers can actually use it also to produce all these different types of short chain fatty acids: acetate, propionate, butyrate, isobutyrate, valuronate, and isovaluronate. So let's look at the sources. So we've got basically, we'll go the one to one. So you've got 4, 10, and 16. All these other ones are pretty high, all these other ones, but the ultra high ones are these 3 that give you, that produce you most. So 10 and 16. So pretty much, what are we looking at? Albumin, that's irrelevant at this stage. And we're looking at 4, which is hydroxyproline. And proline sources, basically a food, will give you more hydroxyproline, so that's very important. And basically serine and glutamate. Okay, so that's for that one there. Propionate 14, and 14 as aspartate. And butyrate, we've basically got again 4, 10, and 16. Again, the same ones, as you can see, like acetate. And isobutyrate, the big one is 8. 8, which is valine. And so isobutyrate, valine, you're actually looking at, which is one of the branched chain amino acids. So obviously, where are you going to find most of it? Beef, chicken, pork, tuna. Obviously, very poor source, and I'll explain later what I mean by that. That's a very source, because you have to remember you've got poor utilization and poor absorbing of this, so it's much lower than that number. And I'll explain later, because I'll do actually the numbers. Go get beans that got bugger all, unless you're eating a kilo of beans to get any amount, and that's going to rip your gut to pieces. So pretty much the animal foods again dominate in this area and always have. So back to this. So that's what the isobutyrate. That's why humans on a species appropriate diet always, if you remember the one that Paul Mason showed very high isobutyrate. For plant eaters, it was practically non-existent. You couldn't even see it, it was so low. So anyway, let's look at these ones: 4, 10, and 16, which is the acetate and the actual butyrate that everybody talks about. So hydroxyproline, which, you know, basically proline plays a role in that as well. And you can actually see even there, in terms of valuronate, is also the proline is playing another role, a massive role in hydroxyproline. So we will look at these, and 10 serine and glutamate. Okay, so where we go. So basically, when you're looking at that, you're looking at gelatin, you're looking at pork, soy isolate. That's pretty much nonsense. you get very little of that, and I'll explain shortly. And cheese, Parmesan, and stuff like that, and more at milk. And this is all 100 grams, which is 3.53 ounces. Now I can tell you protein that is 2.5 scoops. Actually, no, it's not, because they're 30 grams each. So it's actually 3 1/3 scoops, 3 1/3 scoops of basically the sort of protein powders that people put in that regard, because they put 30 grams, you know, so it's about 3 1/3 scoops that most people put in. And most people don't put that amount in the plant-based world. They just don't. So that's just more bullshit. Different day. Then serine, eggs are, yes. Then again, isolates, protein isolates again, soy isolates, which are irrelevant in that regard. And then we go down through fish and stuff like that, cheeses, dairy again, stuff like that. Those sort of things are much, much higher, and most animal foods are in these ones as well. So these are all other manufactured products. Who cares, you know? So that's just manufactured products. Glutamate, soy isolate at the top, again, bullshit. Eggs, you know, seal, you know, good for the Inuits, whale, cheese, cod, meats and other fish, pork skins, they're very high in glutamate. And also gelatin and stuff like that. The connective tissue and all that is very high. They just don't have it here. I don't know why. It usually is at very high levels in the normal when they look at it. But anyway, let me just show you what I mean. So here's a soy isolate. So for this 100 grams, bioavailability for equivalent like cheese, eggs and stuff like that, which is 100%, dairy and all that, and meat is about 98%, you know, around there to 92%, depending on the meat types and all that. But basically we're comparing eggs and dairy here, and to, you know, very close with meat as well. So we will go for the 98, just slightly lower. I'll be generous in that regard. I won't even use the top tier sort of stuff. I'm not even going to use eggs, and we know the bioavailability of eggs and utilization is 49%, nearly half. So much higher. We're going to go with meat, which is 32. So just to keep it fair in that regard. So basically, the calculations is it's about 58% in terms of bioavailability in comparison. So we will divide that with 58, yeah, 98. So we can get a better percentage in terms of relationship to meat. So 59 point whatever. So times that with 17,451. Okay, so that brings it to that level in terms of absorption. Now we have to look at the utilization. All plants and even isolates, plants, but even the isolates that come from plants, it's only 17%. I'm being generous here because animal, they are in many cases between 15 and 17, depending on how they're manufactured. They're purely from plants of 15, but I'm going to give them the top end, which is 17, and that usually is whey proteins and all that at 70% utilization. Well, I'm going to give them the same amount. I'm going to be so generous. You know, I'm such a nice person. Divided by 32, which is meat. So we're not even going with eggs, which is much higher. 48 it'd make this look abysmal. That amount times, which is basically, that is the amount, 5,000, 5 grams, 5.5 grams is basically what it is. That's what you're looking at. really. And that would mean to get the same in, and basically you're looking at, with eggs, for instance, you're looking at double that, okay? Because 11, 4, we're just going to go with the egg sort of product, because equivalent, these are powders for powders in that regard. So, which basically means that you would have to get, because that's 3.33 times, for the same concentrate, if you were getting eggs, times 2, you need to get about 6, nearly 7 scoops. And that's just basically getting close to those limits. Most vegans are probably going to do 1 or 2 of those scoops a day, is about it. 7, it possibly bankrupt them. So as you can see, you can't get the quantities. And it's poorly utilized by the body, unless you're using roids and ramming it into your muscle fibers, which is what a lot of them do in the end. So as you can see, it's piss poor in comparison. Just because you've isolated something and extracted it, in the plant itself, it's much lower. But in an isolate, yes, But that's the only reason that they were able to they can't make any gains, but they can avoid basically full catabolism, which they would get from just eating the plant sources, which have got a whole lot of anti-nutrients. They're bound in certain ways which are less bioavailable for the body because of a whole lot of reasons, that they're not broken down properly. And then on top of that, their gut systems and all that, with the fiber, is completely and utterly stuffed. So they can't get good acidity levels because they've got low taurine levels. So basically that even reduces it, and poor gallbladder functioning because of low taurine, which is in animal foods, unless they supplement taurine to get that working properly so they can actually digest even these poor sources in that regard. So let's get back to this to that. So basically, we can get plenty in the diet in terms of both the butyrate and basically acetate. Now let's take a look at butyrate, isobutyrate. So 8, and that was valid. Where I think, oh yeah, we looked at that. So we've covered that. Now let's look at the other ones. So that's again proline. We've looked at that. So now this one here, isovalenate, is 2, which is leucine. Do I have to really show leucine? I mean, hands down, animal foods are the highest source. I mean, it's a non-brainer, completely non-brainer. It's just basically, forget it. Even dairy, eggs, everything basically just shits all over all these other sources, which have got poor utilization, as I showed you the conversions. So it's not even a discussion, not even a competition. It's like, you feel sorry for them. And 17, which is isoleucine, again. Do I have to say it again? It's going to be basically in animal foods. So and this is the reason why isovalenate and valenate is extremely low in and isobutyrate, again, because valine is very high. This is why they score very poorly in all these 3 type of short chain fatty acids. The ones that they actually get a bit hot, and so 14 there, propionate. So 14, we forgot about that one, and that's aspartate. Okay, so that's the one I actually forgot. I'm not sure whether this has actually got it or not. Aspartic acid. Yep, they've got it in the acid as the, well, And so again, we're looking at soy isolates, then we get into the eggs and stuff like that, then mullocks, pork, again, and more isolates and flours and stuff like that. Yes, you know, if you take it out of soybeans and turn it into an isolate, yes, you can concentrate all these things. You get rid of all the stuff, but actually eating, that's because you've got modern industry to do that. But as a food source, you know, like, look at here, cod, shits all over everything, because none of it, all these other things are isolates and all these other things are flours and everything else. You know, so even the eggs are, you know, egg whites and stuff like that. And then you've basically got, when you're coming to real food, you're looking at mullocks, you know, which is basically crustaceans and stuff like that. Then you're looking at basically, you know, pork skins and stuff like that. And you have to go down a bit until you hit the animal foods, and then the rest is, but it's all these isolates or concentrates and all that. Yeah, fine, but people don't eat that. That's not normal to eat that sort of stuff. But generally speaking, as you can say, so even propionate is higher on an animal-based diet. The only areas where we compete is these two, acetate and butyrate. That's where, depending on your gut microbiome, depending on a whole lot of factors, depending on how much you eat in terms of protein, you know, compared to a vagoon, how much fiber they consume, this is where there'll be a bit of competition, and it can vary between the two. But in all the others, 2, 4 others, key ones, we win hands down. And when you actually look at, you know, so you're looking at, these are millimole concentrations. So you're actually getting well over 400 in the millimole concentrations. So from these sources, plus the other source, so if you calculate them all together, it's going to be much higher. That's what Paul Mason saw in that other more recent study about how on an animal-based diet, the amount of short-chain fatty acid was much higher. And these are small amounts, milligram type. These are small fecal amounts, and they're giving these high numbers. We get more than enough short-chain fatty acids from the small amounts of proteins that end up in the colon. You don't have to worry about short-chain fatty acids, even if you're not in a ketotic state. If you're in a ketotic state, on top of that, you'll get a lot of you know, short-chain fatty. You'll get the ketone bodies, which basically can be converted to can feed the colonic, you know, tissue, as well as short-chain fatty acid. So it's irrelevant. It's an irrelevant argument that is always thrown at us. But I like these old studies that actually are not biased. They're just looking to find out. They're just interested in knowing exactly what are you getting and what happens. Where all the modern stuff is always, you know, so, you know, academia now is so corrupt and so full of vagoons that they're basically always trying to twist, you know, all that, where these people back then were doing pure science just to find out exactly what are the quantities, what is basically created. When we add these things, what sort of levels do we get? This is more reliable research compared to the stuff that they're doing nowadays, where they're just deliberately combining. And a lot of these people are basically like sad dieters who are actually having very poor amounts of protein sources. You know, it's all processed. And then from that, they extrapolate and say, oh, this person is a meat eater. They don't use the word carnivore. They use meat eater, even though they're only consuming about 30% of their diet from meat. And all the other ones are refined sugars. Now, that's a meat eater, and they have very poor short-chain fatty acids, where the vagoon, who's eating a whole lot of kibble, a whole lot of fiber, oh, yeah, they got, you know, it's nonsense, absolute nonsense. You know, if you're on a species-appropriate diet, you get more than enough, let alone the ketone bodies. So the studies, the way they're done nowadays, they're not representative. It's all these sort of reviews upon reviews upon reviews, and they're all basically on sad dieters not on basically looking at, removing all that other stuff, just putting in the proteins and seeing what happens, and really getting real, you got the bugs, you got the small amount of proteins in milligrams, and what are you getting from that small amount that would end up, because you won't digest every single bit of protein. Some of that in small amounts of milligrams and all that will end up in the colon, and then it get excreted. Even we shit, as carnivores, but it's much less the quantity in that regard. But even in those small quantities, as you saw, you get more than enough in terms of short chain fatty acids to basically provide the colonic cells. And if you're in a ketotic state, because you're having one meal a day and you're swinging in and out, you know what? In this low-grade ketosis, you're going to be feeding with ketones those bugs anyway. So, not going to have an issue. So don't worry about it. Research backs you up in that regard. And if, let's think about it. If this is so important, why weren't the Inuit or the Maasai, all these multiple generations, do I need the research to basically prove that they are healthy and they got healthy guts and healthy gut microbiomes? They got way more healthier than ours, and we know that from research. When we've looked at their gut microbiome, is much richer than ours. So, and they don't have any of these IBS issues or issues that we have in our modern world. So they get more than enough from the proteins that they consume. Now, obviously, if you're consuming like a very poor, you're doing one of these strange therapeutic ketogenic diets where you're just relying on ketones and you basically have very poor protein intake. Could that create an issue? Maybe. I don't know, because we've never researched it. But it's probably contraindicated. I would say what is indicated is basically to eat a species appropriate diet. And when you look at tribal populations, they maximize the amount of meat and fat they put it down their throat. So they don't spare anything. It's all food. Put it away. They don't say, I can't eat that amount of protein. I can't eat, or the other nuts. I can't eat that amount of fat. No, they eat the fat with the meat, and they gorge on it. So be a proper carny. Stop basically being a little bee. Can't use that word just in case it affects the YouTuber. You know what I mean, the bee thing. A little bee, exactly. Anyway, thanks for watching. See yous.